welcome to yet another micro seminar. We're really excited to be broadcasting not only through YouTube to the entire world, but also down to Texas for ASMQ. And we're very excited to have Jason with us today to be talking about fungi and comparative genomics. And Jason, we can take it away, and he is out at University of California, Riverside, right?
Yes, that's right. Thanks, Jen.
All right, so Jason, take it away.
Okay, thanks for the invitation. All right, I will now hide behind my slides, I think, and I will share my screen.
And while you're sharing your screen, I will just remind everyone that to ask questions, what we're going to do is have questions come through Twitter. You can use hashtag #USeminar or tweet them to microseminar. We'll try to follow the ASMQ hashtag also in case you forget and you tweet it to ASMQ. And if you're in the Hangout, please stay and ask questions, but we might actually open it up to ASMQ for questions first.
All right. I can't see any of your facial expressions, so if anything happens, you'll have to speak up since I'm only seeing my slides. But thanks for coming, and I will talk to you about some projects that we are doing to study fungal genomes and how to understand fungal evolution using these tools. And I put a bunch of links up here for some of the things in our lab, and I'll point to these later. Because I want to first start off with just an emphasis on the important things that fungi do in the environment. Here's some illustrating some cases of fungi associating with plants. On the left-hand side are some cultures of different endophytes that have been grown out of leaf tissue, and this is from Betsy Arnold's lab at University of Arizona, where this is just showing the remarkable diversity of morphology and color and in many cases metabolism that you can find just in fungi that are growing inside of plants, and they're just they play an important, maybe particularly underappreciated role in some of the ways that plants deal with their environment. We are probably more familiar with some of the mycorrhizal associations, which is at the bottom right here, showing the mycelia that associate with the roots, and they provide this communication and with the plant to be able to share resources, in which the fungus gets sugar resources and the plant gets usually nitrogen resources back up from the fungus. So there are these intricate partnerships, and then I'm showing you here just some other pictures of fungi just to emphasize that there's a lot of different diverse forms of fungi. The really big picture is a picture of a Pilobolus fungus, which is a dung fungus, and those little black tips there are spores that will be ejected, and I decided not to put the video up, but there's some great videos from Nick Money's lab and others of these spores being ejected, and the incredible force that's used to allow the spores to escape this dung pile and to make it probably into the grass or elsewhere to be picked up and either passage through a ruminant or blown into another dung pile. So there's a whole interesting aspects of their biology, and so these are interesting to me is there's lots of different ways that these organisms have evolved to spread their spores. You can see on the top right you have mushrooms, and again that's a specialized multicellular structure that is formed for sexual reproduction, and it's the height of the mushroom in some cases is to enable spores to be wind dispersed, while other ones there's actually specific plans for digestion of the mushroom so that the spores can be more can be spread in a different way. So there's really interesting differences among the different species within the kingdom, and I think that's quite interesting. And so what we're driving at is how we can understand these processes, how these aspects evolved. The bottom right is just a picture of a mole cricket, which is the collaborative project I'm actually working with Adler Dillman, who's a professor here at UC Riverside, on looking at some fungi that are able to kill these crickets, which are invasive, and we're just interested in studying it. But it's interesting just to remind you again. Here's this intricate association, but the little white tufts that you can see are the fungus kind of starting to poke its way out of this dead cricket. So, anyways, lots of cool, I think, associations. And so what I think that what we're really driving at is can we understand these associations and the roles that they play in terms of how the genome is shaped by the differences that these organisms have in their ecology, in their morphology, and their metabolism. So we're going to focus a bit on early divergent fungi, which is I'll define in a second, but are some of the early branches in the fungal tree, in part because I feel like they've not been studied as well with the genomic tools, and we're just now getting these resources. And but I also think that they're really important samples to study what might have been the ancestral states of the fungi. So being able to study the early diverging lineages gives you sometimes can give you clues into what the earliest ancestor of the fungi might have been doing, and that's really interesting to me, and trying to understand how that relates to the really amazing amount of diversity we see in the kingdom. What did the ancestor look like, and how different are the things we see today from that? So we can observe some of the transitions and growth forms across the kingdom, and see how these correlations might how these changes might correlate with geological time. And so, for example, when fungi move from being mostly aquatic to terrestrial, can we observe the changes in the genome or in the gene sets that might relate to that? We can also look at differences between things that are free living, saprotrophs, versus ones that are associated with plants or animals, and how that shift, how so fungi were around before there were plants and animals, and so when plants and animals became well, when they evolved and when they became more dominant on Earth, what changed in terms of the fungal repertoire of metabolism? So these are interesting questions, and we think that genomics can provide some ways to try to understand that. So that's kind of the driving purpose of some of these studies. And so now I'm going to talk you through just some of the resources that we have. This is just one schematic tree of the fungi. If you remember from any of your micro or organismal biology classes, the fungi are broken into several major phyla. The definition of some of the phyla for the early ones are shown here in this comb-like structure, because at the time we really didn't know exactly what was the earliest branching for some of these. So they're listed out here: Mucormycotina, Entomoptera mycotina, and so forth. But I'm showing you on the fungi that you might be most familiar with would be things like mushrooms, and they fall within the Basidiomycetes under the Agaricum mycotina. I can't point with my mouse, but the Ascomycetes are where we would find things like Saccharomyces that are very good at helping make some of the beer that some of our listeners are drinking now, and as well as the filamentous fungi and the Physiomycotina. Again, if you have an orange in the fridge or on your counter that's starting to get a white or a green mold on it, that's usually a filamentous Physiomycotina. So anyways, across the kingdom there's been several innovations, and I'm mapping on some of those in terms of things like when the flagella was lost, and we'll talk in detail about that, and then some other sort of structures that have occurred when things like regular septa or changes in growth forms. And at the time of this paper, we presented the idea that there were at least a couple of origins of multicellularity, and so we're interested in understanding what changed in fungi to allow for these more complex forms to arise. And it's probably the case that there are more examples of multicellularity, but these are some of the major ones that we think were pretty remarkable to kind of mark on the tree. Okay, so in terms of resources, there are more than 400 genomes of fungi that have been sequenced to date, and I'll speak in just a second about our efforts to do that, but this is just kind of a snapshot of the genome sizes of about 350 of those genomes, and so you can see that there's some outliers that are at the high end, and those tend to be things that are that contain a lot of transposable elements. A lot of times some of the plant pathogens are also tend to have a large a high number of transposable elements, and then you have some of the genomes that are extremely reduced, like Microsporidia and some of the yeast, which will be Microsporidia is very reduced. They're only about 2 megabases at the smallest end, and some of the yeasts are in the sort of the lower end of the 8 to 12 megabase range. So we see variation in size, and one question, of course, would be, does complexity correlate with that? And we have one example we test that I'll get to in the talk. So to generate these data, of course, there's lots of different efforts that have gone on. The Broad Institute has sequenced a large number of fungal genomes through the Fungal Genome Initiative, and then the DOE's Joint Genome Institute has been an incredible partner in sequencing more genomes, and so through several different aspects of their community sequencing project, in a project that we started a couple years with them years ago has been to try to bring the number of sequenced fungal genomes up to 1,000. And so we called this I called this the Noah's Ark of fungi, where we were going to get 2 genomes from all of the 500 families of fungi as a way to kind of make sure we're sampling the phylogenetic diversity of the kingdom. As we dig deeper into these samples, we realize that we don't know that much about the diversity, and so 500 is perhaps a it's an arbitrary number for families. There's probably quite a few more as we start digging in deeper. But of course, to do this, we need to have cultures of these organisms, or we're doing it now from fresh live tissue. So there's been different approaches that have been tackled with this, and we're making progress. To date, probably about 300 genomes of these samples have been generated, and I would say there's probably another 100 that are in the pipeline through different stages. And again, our goal is to enable the 1,000 genomes. It doesn't have to be all done by us, but we would like to see this happen through efforts across the world. And then, of course, it's what do you do with this data once you have it? Just having the genome isn't sufficient, and so building databases for this is another aspect. So inspired by Jen, I made sure I put a video together to remind us about. So if you haven't seen the preview of my talk, this isn't quite as funny, but I just want to remind us that not that Knight Rider necessarily knows how to deal with genomes, but to remind us that there can be a lot of chaos when we have all of those, all of that data, and how to kind of process it. And so one tool that my lab, in collaboration with the UPathDB project, has developed is called FungalDB, which is a place for being able to interact with these genome data in a way that allows you to build reproducible pipelines for queries. And so it's not just about browsing the genomes, but actually being able to ask questions. And I don't have time to go through it in detail, but the idea being that you can build these steps, like you can see at the top, and allows you to build things like identify genes that are differentially expressed through some experiments, but then also find their orthologs and map those to different in different ways. And so, anyways, this is just a snapshot of the interface, but the idea is to be able to generate these gene lists that come out of these queries, and then build these pipelines that we can then save and actually rerun as new data come about, or share with collaborators. So again, I have no idea if my videos are funny or not, but I'll keep going since I can't hear you guys. But I think that it's important that we deal with these data.
Would you please repeat the question?
Is that we make sure that we understand that that we have to think about how we're going to deal with the data before it becomes overwhelming. So that's one of the reasons why these databases are there. Okay, you appreciated it. That's good. Thanks, Jen. Okay, so to keep moving on to the results, I want to just emphasize the things that I think we've learned about fungi, having had the perspective of several years of molecular phylogenetics, is that morphology has not been sufficient to resolve how fungi are related. It's been good for major groupings, but there's a lot of microfungi where it's very difficult to tell really what they are. And molecular phylogenetics has incredibly transformed mycology, and I think that genomics is this next wave of how we interact with mycology. And so the phylogeny that we're able to infer from more than just a few loci has become even more robust with this kind of data, and able to solve some of the problems that we've had with single gene phylogenies. But also we're able to look at things like the genome characteristics as a trait and see how that changes over time. And so these are important things, as I alluded to, when we're trying to understand how metabolism changes. We can use the genomes to go for that. So I'll just try to speed through a little bit, just a few more of these things, but just to emphasize that one of the first publications that described PCR primers for how to amplify the ribosomal locus from fungi, which transformed how we did molecular phylogeny in fungi, has been cited 15,000 times, which is quite a lot for a paper, especially since it's a book chapter. But this has really transformed our ability to understand fungi, the relationships of fungi. Another kind of important point in that was this development of this molecular phylogeny from 6 genes that was published in 2006 that described really really got the relationships much more nailed down for a lot of the major groups. And so one of the interesting questions that came out of this work was, what is the earliest branching fungus? And so down here at the bottom you can see this fungus called Rosella alomyces, and that was inferred to be one of the earliest branching fungi. You can see sister to it is this group called Microsporidia, which had been problematic in terms of figuring out where they were where they fit in the fungal phylogeny, and they'd actually bounced around to several places on the tree. But this tree presented the idea that, as well as others, presented the idea that they were sister to Rosella. But there was not that much support for that because, in part, the Rosella have really long branches, and so it'd been difficult to be confident about that. Also, recently, what was discovered through sequencing 18S sequences from both freshwater and aquatic sediment was this novel, diverse group here. And so if you look on the right-hand side, you can see this tree where the blue arrow is a little bit down, but it's supposed to be pointing to Rosella. And you can see that up above that, there's a whole bunch of these clone IDs that are listed. And these were all novel clones that, when they looked at them in a phylogenetic context, realized they represent this whole other clade of fungi. And so the group from Tom Richards' lab at Exeter named this the Cryptomycota, and they were able to develop probes to go back and probe the samples to actually identify the cells where these sequences were coming from, and show that they likely had characteristics that we see in some of the chytrid fungi, which is that they have these flagellated stages called zoospores, and that they represent a whole lot of diversity that we had not seen before or not really characterized. And so one of the questions that came out of this was, well, that's just from one gene, and how confident are we that this Cryptomycota and the Rosella are really sister lineages, and where do Microsporidia fit? And so again, Rosella is this intracellular parasite of another chytrid fungus called Allomyces. So Rosella is in this label F here, are these spores that look kind of spiky, and they're inside another fungus. And so that is this. They're a mycoparasite, and we think they're this earliest branch. And also Microsporidia, if you hadn't seen what Microsporidia look like, there's this long. They have this very intricate apparatus called the polar tube, which is sort of like a harpoon that they throw out, and they're able to hit a cell and then inject their contents into the host cell and begin part of their life cycle. And so so that doesn't look like a filamentous fungus at all, right? So that's another reason why, or really like a yeast. And so one of the reasons why it was hard to classify Microsporidia as fungi is they really looked so different. So through lots of genomic approaches, and I'm just going to summarize all of this in one tree, we sequenced the genome of Rosella, and we're able to resolve the placement of it with higher confidence, because we're able to use about 200 protein coding genes, and we can find really strong support for Rosella and Microsporidia to be sister species, more so than was able to be shown in the previous the previous 6-gene phylogeny, and also get this placement of these fungi as the earliest branch. So from this work, we were able to say for sure that that Rosella is sister to what we call the Cryptomycota, and there's efforts underway to get some of those other Cryptomycota, which we only have 18 S's for, actually sequenced from genomes. So with single-celled approaches. So there's very interesting things going on in this this realm. So just to keep going forward, some of the other branches that were really difficult to resolve, which was that comb picture that you saw when I first showed sort of a schematic. We've been working to try to resolve that comb into a better relationship, and other as well as others have been working on this. But through using, of course, genome sequencing, we're able to take those clades, which you can see on the right-hand side, which I'm sure nobody remembers the names that you saw before, but those were most of the same clades as were in before, and we're able to resolve aspects of their relationship. So, for example, the glomeromycota, Morchella mycotina, and mucormycotina, those are the names they currently have for those groups. Those are all more closely related to each other and are sister to the ascomycetes and basidiomycetes, and then we have the other group that's the Coxella and Entomoptera mycota are kind of an earlier branch. So we're able to do these kinds of things because we have more sequencing data, so more sampling, but we are identifying cases where there's conflicting signal within the data, and so some of these conflicting conflicts may be due to horizontal gene transfer, perhaps, or they may be due to incomplete lineage sorting or differential rates of evolution. We're still trying to really sort that out. But the order, for example, of these two branches, the Blastocladiomycota and the Chytridiomycota over on the right-hand side, the order of those two has been very difficult to pin down, and so different phylogenies, different analyses resolve them in different order. So there's still open questions that we're trying to tackle by both adding more samples and improving the models that we use. So one thing that came out of the study of the molecular phylogeny was taking that tree of the fungi and mapping onto it traits. And so there's many interesting traits about fungi, but one that's interesting that I didn't fully emphasize here is that early diverging fungi have a flagella. So they have a zoosporic stage where they swim around, and if you look at this tree, the little circle with the squiggle is supposed to represent organisms that have a stage, and a life cycle stage which includes a flagella. And so the inference from this tree, because of the losses and gains of the flagella and the phylogenetic relationships, was that there were at least 3 losses of flagella in the fungi. And so that's what the tree at the time is able to tell us. And so we'd like to be able to revisit that with better confidence in those relationships of the fungi. One of the most difficult ones here is this species called Opidium, which unfortunately, we've had a lot of trouble getting the genome sequencing completed for that, but that one is going to be very important to be able to identify it sort of in the middle of your screen here, because its sister species does not have a flagella, and the related sort of sister clades also do not. So what does that mean? It was retained and lost multiple times, or is it placed in the wrong place? So, anyways, these are going to be important questions. But and I'm going to sort of tackle one part of this in just a second. One more intro here is that another thing that's really interesting about the differences. So you have the flagella, which is this important characteristic that was lost in what we would think of as the terrestrial fungi that make filaments. We only see it in the fungi that have these zoosporic stage. But also is how they do nuclear division, and so the important part of segregating your chromosomes is to be able to have a microtubule that can attach to them and pull your chromosomes apart as part of division. And there's an attachment point on either side of the cell to be able to enable that. And in animals, that attachment point is called a centriole. And you can look down here at the left-hand side, and you see this cross-section of an animal centriole, which is also related to the flagellar. Some of the similar proteins are involved in terms of the flagellar apparatus and the basal body. Those are all related here. But on the right-hand side, there are proteins that are involved that are implicated in that. On the right-hand side, you can see what's called a spindle pole body. So it doesn't look at all the same, but these are not homologous structures, but they perform analogous functions. And so a really important question is, how did fungi, well, what do fungi do, and how other fungi do? And so if you look at the chytrids and look at this cross-section, you also see a chytrid centriole looks very much like an animal centriole. So the hypothesis would be that the proteins that are needed to be for that are also found in the genomes of the chytrids, but are presumably not found in the fungi that don't make these. And so we can profile that and try to ask what's what's conserved. And so actually aspects of this were addressed in a previous paper from Scott Dawson's lab and Zach Handy's lab at Berkeley and Davis, as well as many collaborators, where they sequenced this organism called Negalaria, which is an interesting branch in the eukaryotic tree where they have both an amoeboid and a flagellar form. And again, I'm just, I don't want to go too far afield here, but the important thing was that they were able to identify genes that were found in flagellated organisms, and they build these profiles of these genes. And so they have this nice pattern of genes that are found in only the flagellated species and missing in the ones that don't make flagella. And so it's nice. There's a nice inventory of genes that we can look for. And so what I'm highlighting is the fact that the only fungus that they were able to really survey, they didn't have any flagellated fungi that they could survey at the time, so they weren't really able to incorporate that into the figure. So here's an updated figure from my work, looking at many of these FM proteins that are on the left-hand side, and so all the fungi, all the organisms that have a flagella are highlighted with this greenish color, and you can see that they all share most of these proteins that the previous publication argued are important for flagellar motility. They're all found in all of these ones that have a flagella, and then you can see that some of those include fungi, but then there's a break, and all the fungi that don't make flagella don't have these genes. So that's a nice kind of confirmation that we can take a molecular character and look at the genome and find the genes, find out an assemblage of genes that are either present or absent. And so we can ask other questions like this: are there other genes that fall in the same category that behave in the same way? And so I've taken a larger sampling of some of the flagellar motility proteins that were identified, and we can look again at this sort of structure of flagellated versus not, and identify things that are shared. And so not everything is, of course, perfectly conserved as that smaller set, but there are nice patterns of conservation. And so these are essentially the things that were lost as fungi, presumably as they lost their flagella. And so again, sampling more taxa, we'll be able to get a little bit better handle on exactly what happened, or we hope, because my questions are, which I think I'm going to summarize in a second, but did it happen? Did all of them have to be lost at once? And here's just another sort of breakdown of these in terms of what some of these genes do and their kind of presence absence. So some genes are, of course, found all the way across the board in terms of in fungi, but and because they must have homologs that are either preserved or have other functions and weren't lost. But then there are nice examples of things that are missing. So I think the open questions and the way that we're going forward with this is sort of what I like to think of as kind of the first stages of doing evolutionary cell biology is trying to understand if all of these genes were lost at once. Are there genes that are still retained that may have acquired other functions? Is this a sharp gradient of loss, or are there sort of things? Is it a sharp cutoff and sort of lose flagella, lose every protein, or are there some things that are still there? And is there anything that's also gained in the context of the loss of the flagella? So we can identify other proteins that are found only in the fungi that don't have flagella, and so those might be interesting things to look at. It's sort of in the opposite version of that pattern in the heat map. So this is just one way we're trying to tackle it. I can say we're only at early stages. We don't have a lot of molecular tools to manipulate these early diverging fungi, but it's something that we're actually actively working on, and so we hope that can get us closer to that. Sort of tackling this question in another direction is sort of taking the idea of complexity on and asking, in addition to the flagella, are there other characteristics that have changed? And so one thing I alluded to before was this idea of complex multicellular structures. And so we see these are really important fruiting bodies for sexual reproduction and dispersing spores, and we can identify through comparative genomics some of the patterns of genes that are found, not unlike that previous pattern, genes that are found in things that have complex multicellularity, and then genes that are missing and ones that are more simple. And so this is just a movie of a fungus, Neurospora crassa, growing, and you can see that it's the important thing I want to emphasize is that there's a lot of turgor pressure within this hyphae. So this is a hyphae. There's pressure within there, and that pressure and this polar growth that pressure is supporting this polar growth, and so it's important for the cell to be able to to support this pressure and deal with it. And so, which I'm going to explain, sorry, in a second. But what I just want to sort of remind you of, this is just a smaller snapshot of not the early diverging fungi, but some of the other fungi, and we think that there's probably examples of some of these multicellular forms with these complex fruiting bodies across the board. And these are just some eye candy of some different fungi from different parts of the tree. I don't expect you to digest too much from this, but just to say that some of those have kind of complex multicellular forms. Some of them grow as single cells. These are some fungi that actually make truffles down here at the bottom, or have these other complex fruiting structures. And then we also have within this group that's related to Saccharomyces pombe, we have a lot of yeasts. But what's interesting is that within the group that we call Tephrinomycetina, there's also one lineage that has an interesting form, which is that it actually grows in a multicellular form. So all of its relatives in that whole group, that whole clade, are single celled, but we have Neolecta, which has this, we call it sort of like an earth tongue, but it makes these fruiting bodies that are quite complex. And so, sorry, to go back to what I was saying about the pressure. So in order to deal with that pressure in the cell, the important thing happens is what happens if the cell gets broken. So if you get cut by a razor blade on a petri dish, but also if you have a big water droplet that hits you, or something kind of chomps on you, your hyphae can break, and without any way to stop the bleeding, you will essentially lose all of your cytoplasm, and the organism will die. And so organisms have developed these things that we call septal plugs, or Ronin bodies in different groups, but they're basically these septal pore plugging proteins that are important for this. And sort of like when in a submarine you have different hatches, it's the same idea. You want to close a hatch if there's a rupture, so that at least not everything leaks out. And so there's examples of these septal pluggings in both the filamentous ascomycetes and the basidiomycetes, and that's shown here on the tree, where there's the SPC at the top is the septal pore cap, and that's one of those kinds of pluggings that's found in one group of fungi. And then the WB at the bottom is the Ronin body, which is another kind of septal plugging. And what we've learned by protein sequencing and purification of these proteins is that they're totally independent proteins. They're not homologous sequences, and they've independently evolved this ability to plug the holes. Okay, so the important question is, well, what about Neolecta? So Neolecta is on that same branch as Saccharomyces pombe here. How does it deal with the fact that it grows as a filamentous fungus? It has hyphae. What does it do to deal with those plugs? And so Neolecta is kind of a mysterious organism. It actually wasn't clear where it fit phylogenetically. So some previous work had sort of puts it within relatives of the Saccharomyces, but its placement had been quite confusing, and it was called sort of a fungal dinosaur because it might represent kind of a really old early branch in that group. And so again, to solve this problem, although I guess I've already shown you a tree of where it fits in our data, but is of course we use our favorite tool for things like this, which is genome sequencing. And so this is just a summary of genome sequences from Neolecta in the very left-hand corner, left-hand column, and then several other genomes that were sequenced that are in the same group. And so the important thing is you can see that the genome sizes vary a little bit, but in general, the genome size for Neolecta is not that much different than the other organisms. The gene count number is in the same ballpark as the other ones. And so at first glance, even though this organism has a more complicated lifestyle and morphology, it doesn't have a dramatically larger number of genes. That's the first observation. If we go back and sort of revisit the phylogeny, we get this placement of it likely to be as one of the early branches in the Tephrinomycetina, and we can place it there pretty confidently with this new data. We still have some uncertainty about everything in this Tephrinomycetina group, but we feel okay that it's probably pretty early. And so if we go in and look at the Neolecta under EM, we can identify these septal plugs that are occurring. So we can say that Neolecta makes these septal plugs as well. And so we can ask the question, so we know the proteins that are involved in what are called the Waronin bodies in filamentous fungi, the filamentous ascomycete fungi like Aspergillus or Neurospora. We know those proteins. Do those proteins exist in the genome of Neolecta, or does it have a different way of making these plugs? And so to do this, at the top we took genes that are necessary for making these Waronin bodies. They're part of a set of genes that are called the hex proteins, and they the hex proteins are called hex because they if you purify the protein or make the protein, it actually self-assembles into these hexagonal crystals. So it's this very and energetically, it has this really interesting properties of sort of aggregating. And so you can see in all the red lineages at the top, these are all ascomycete fungi and you can see the little WB at the left-hand side of that tree, part of the tree. That shows when we think the Waronin body originated, because all of these species below it all have the hex protein and lichen and whisk, which are necessary aspects of decorating the Waronin body. They all have that protein, whereas we do not see any copies of that if you see the NI lineage on the tree kind of in the middle of the screen, that even though it makes these plugs, it does not have this protein. So that suggests that this protein that makes the plugs is independently evolved. And so to try to answer this question even more, again, this is work from Greg Jed, who's at TLL in Singapore. He purified proteins from because in general, Waronin bodies are really highly expressed proteins. He purified some of these, and we tried to sequence them and identify the sequences, and to see if we could find anything that looked like it might represent that protein. Unfortunately, even though we got some bands and were able to sequence them, we didn't find anything that suggested to us that it was actually a Waronin body. But we do have lots of highly expressed genes from an RNA-seq experiment, and so we're still thinking about ways to maybe match a potential gene to that process. So one last thing about this idea of complexity is if we take the Neolecta genome and we look at what percentage of the genome are found in other fungi. On the very right-hand side, you see this number that there's a red box, and it says 1210. That represents the number of well, in that case, isoforms. If we reduce the isoforms down, it's about 1,000 genes, 1,100 genes that are specific to Neolecta. So those are genes that are only found in Neolecta and are not found in any other fungus. And so those potentially, if something was acquired, if some sort of adaptation and novelty occurred to be able to make Neolecta, for Neolecta to support this multicellular growth, maybe some of these genes are important in that process. And so that's what we can find. About 22% of the genome is unique, whereas more than half the genome is shared with all eukaryotes, and even more like 60% of the genome is shared with other fungi and opisticons. So there isn't necessarily a huge amount of novelty in this genome, even though it seems to have novel form. So that's kind of the take-home right now of what we've been able to discover based on the genome sequencing and comparisons. And again, this organism isn't tractably genetically, so we don't have a way to go in and specifically test individual genes except by cloning them out and trying to test their function outside. So that's kind of a whirlwind of a couple of things there on some complexity and using the genome to kind of dig down. I want to just go into one other study on early diverging fungi. As I said, this was going to be a survey of a couple of different studies, and I apologize for those that are maybe overwhelmed by the different fungi names and groups, but hopefully you get a sense of some of the tactics that we're using. And so we want to ask some questions about cell walls and metabolism for an emerging fungal pathogen. And I want to study cell walls because I think they're one of the unique aspects of fungi that distinguish them from a lot of their hosts, is that they have this composition that's different. Is made up differently than what you see in plants or animals. And so we're going to talk mainly about this fungus called Batrachochytrium dendrobatidis. It's a major cause of amphibian decline. It's a zoosporic fungi. It's a chytrid. So it has this life cycle where it grows as a zoospore. That zoospore finds a place to land. It eats its zoospore basically, and it encysts and develops into what's called a zoosporangia, and then within that zoosporangia the cells divide mitotically to produce new zoospores. And so this is the major reason why we're seeing amphibian decline. It's a huge problem in the Sierras here out west, but also in all over the world we're seeing amphibian decline BD is causing a major problem. When you look at BD under the microscope and you look at its interaction with the cells, it's actually on the right-hand side. You can see the BD cell is the circular thing, and on the right-hand side is the frog cell. It has this long association with the host cell. It's interacting with the host, and so I think this is important for how it might be communicating, or what kind of molecules is it sending over to the host to infect it. There is, in fact, another species of Batrachochytrium that's been described called Batrachochytrium salamander, which is a pathogen of salamanders. It actually has a different pathology in that it's more necrotrophic, whereas BD is actually more of a longer-term exposure, causing the skin to slough off, and more of an osmotic problem, regulation problem. This BS is basically causing actually more cell death immediately. So there's a different pathology there. But I'll just focus on BD because that's what we've done our work on. So in about 2004 or 5, there were 2006 or 2007, there were about 2 genomes of BD that were sequenced both at the Broad Institute and JGI. And what we did, again, what do we do when we have genomes? Well, we try to compare them. And so we asked some questions about what were different in the BD genomes relative to some of these other chytrid genomes which we had access to. And I'm just listing at the bottom some of the gene families that appeared to be quite large in BD, but weren't necessarily that large in some of the relatives. And so we tried to highlight these and try to understand, are these important aspects to pathogenesis? But I wanted to, remind ourselves that seeing big families is nice, but doesn't necessarily mean that the changes are related to adaptation. And so we wanted to identify a closer species that might be able to help us, because the branches are really, the other samples are quite far away evolutionarily. And so to do that, we went and sequenced another fungus called Homoflipping polyryzza, which, again, I know that these are names that are new to everyone, I'm sure, but just another species that we could identify that was the closest thing to BD. We sequenced that genome and annotated it, and Suzanne Johnson, who was a postdoc at Idaho at the time, and Erica Rosenblum's lab led this effort and was able to first identify, of course, that it was closely related, and then we went back to these genome annotations and it identified whether or not the gene families that we think were large in BD were still, in fact, large when we compared it to the close relative. And you can see for some of these species, some of these families, like the tyrosinaceae, the S got cut off, but that it was not significant. That wasn't really that much larger in BD than its relative. So we kind of discounted that one for the future study. But basically, we took these families, and we realized that the M36 family are, in fact, what are called fungal lyosins, which are known to be keratinases, known to break down keratin, which is in the amphibian skin. So that's a really interesting family to find expanded. And we also found other serine proteases that were expanded, and interesting to us was that some other serine proteases were found to be expanded in several other animal pathogenic fungi like coccidioides, and so it was just curious to find the same family continuing to expand independently in different organisms. So to really get our confidence in this expansion, we built phylogenetic trees for the families. And all you need to know here is that the black bars are the BD copies. And what you can see is, even without sort of figuring out what else is going on, the black stuff clusters. And so that's an indication of that it's a recent expansion, because they're all most closely related to the same species. And so we're able to both identify that the expansions are recent, because the number of synonymous changes was quite small for some of them, and so that really indicated that this was a recent duplication of these families. And we see this again for M36s, the S41s on the right, and also another family of aspartyl proteases was also extremely expanded in BD relative to the others. And so those are quite interesting to suggest they may be important in pathogenesis. Again, without a transformation system or ability to knock things out, we can't directly test that yet, but these are observations we can make. We were able to look at the M36 family and its gene expression differences, and we found that most of these M36 families were upregulated in what was called the Sporangia phase, and that's the phase when it's living in association with the frog, when it's sitting down on the frog. And so that would make sense if it's sitting there secreting some sort of protease that's helping digest the frog, so it's able to absorb those nutrients. It's kind of logical to think that it might be upregulated under that stage. One final experiment we did with that was to actually take HP and BD and grow them and incubate them with frog skin. And you can see on the left that HP does not change is not able to degrade this frog skin at all after 2 or 3 weeks of being in that culture, whereas BD has left behind this sort of soupy broth that is the remains of that frog skin. So again, we can't directly implicate those proteins yet, but I think those are experiments we're going to try to get a better handle on. But it seems like this difference in the proteases could explain some of the necessary steps for pathogenesis. So that's another kind of comparative study from that. One other observation, if you remember, there was one other family in there that was large called CBM18s, and so a postdoc in my lab went into this family and wanted to look a little bit more carefully at it, and he identified that these multiple copies of the family, which is what you see here with the BDEG and the gene tree on the left. But what he also identified was that within the genes, they had multiple copies of this domain. So it wasn't just that this domain was that the gene was multi-copied, but the domain itself had many copies. And so we classified them into different groups based on the secondary domain that we see present, this deacetylase or tyrosinase-like gene. And together we're able to to identify all of these copies, and we put letters on them based on building a phylogenetic tree of cutting out each domain and building a tree. And what we found was that all of the lectin-like domains end up clustering with each other. That's the red clade. And so we think that most of the domains are evolving from tandem duplications. So it's really interesting because most fungi don't have that many copies of this domain, and yet it's extremely expanded within this species. And many of the copies appear to be evolving under rapid positive selection. So we were really curious to think about what this could mean. So it's the largest copy number of CBM18 and almost all largest. This copy number is the most in almost any fungus, and one gene has 11 copies of the domain, and that it's thought to bind chitin. And so maybe it's binding its own chitin, or maybe it's binding in a way to cloak the cells from the host immunity. So if you hide your chitin is one of the distinguishing features of the fungal cell wall. Maybe the receptors in the animals can't, or the host can't recognize it, and so that's a possibility. And in fact, this has been shown in plant pathogens that chitin binding can protect a cell from the host response. In this case, it's able to prevent the host's chitinases from being able to attack the fungus. Okay, so that's one study that had been done in plant pathogens, and so another postdoc in the lab wanted to know whether or not CBM18 could really provide this role of protecting the cells from chitinases. And so we cloned out several. She cloned out several copies of the gene. We didn't do all 11, but she took several copies and we expressed them in yeast and tried to see where they localized to. And so a couple copies did appear to localize to the outside of the yeast cells based on these, and some of the other ones, or these didn't seem to localize with other known structures within the yeast, and so but they have a secretion signal, and again, we're taking something that's probably 1 billion years. It probably has diverged for about 1 billion years between yeast and Batrachochytrium, and we could express that protein in yeast without any trouble really, which that was kind of satisfying. And so then Peng took these copies. She actually engineered versions that were either the full-length copy or either just, she called them MA and MD because the letters that we'd assigned to those domains were either A, B, C, D, and so forth. So there was a copy that had 3 A domains and 3 D domains. So she made either versions of those, and then just a single version of the domain. So she made these constructs and expressed them in E. coli. We purified these proteins, and then we asked whether or not these proteins could bind chitin. And so here we do this assay where we incubate them with either chitin or crab chitin or cellulose and xylan. And what you can see is that in all cases, either the whole protein, which didn't express particularly well, so that's why the partial copies did a little bit better. We get basically specific binding to the chitin, but not to the cellulose or xylan, and we use the AVR4 protein as a control in the bottom right here because that was also been shown to have these same properties. So we're able to say that that this gene this protein is able to bind chitin, and the other question was whether or not this chitin binding was important for protection of the cell. And so in AVR4, the experiment that was done is to take a fungus called trichoderma, which is very sensitive to chitinases, and that's what you see here in the A is just trichoderma growing in liquid, and so you get this fuzzy stuff. If you add a chitinase, which is what you see in B, you see all the fuzzy stuff goes away. If you add back in, if you incubate the trichoderma with AVR4, as you see in C, and then you add the chitinase, you get protection, and you still see growth of the trichoderma. And we see the same thing for our MA and MD domains. And of course, with an empty vector, you still see loss of you still see degradation of the host. So this is nice evidence to suggest that this protein can also provide this protective effect against chitinases. So we think that. Overall, some of these studies in the BD genome has been able to tell us that there are changes in the metabolism that might be changes with shifts to a host, because none of the other relatives of BD that we had sequences for associate with animal hosts, but that these changes perhaps in the metalloproteases may be important for how BD has evolved to become a pathogen, and we'd like to understand that better. The CBM18 gene might be an important protective gene that can bind chitin and could play a role in its ability to be virulent. So with that, I know I talk really fast, but I did get through all the slides I had prepared. This is, instead of a mushroom, it's a mushroom cloud to go with Jen's exploding preview, and I'll just stop and thank the members of my lab that have contributed to this. Peng was instrumental in doing the CBM18. She led that whole project, all the expression, and John was a previous postdoc who did the computational work. Usmani is a postdoc who's worked on the Neolecta project and is an expert in doing the comparative genomics work. Stephen and Divya also contributed a lot to this effort. There's a lot of collaborators on the sequencing projects and some other things that I didn't quite get a chance to talk about. But I threw some URLs up, and I think I'll stop there. So there's at least a few minutes for questions if people want to.
All right, thanks, Jason. I'm trying to get my text to play here. My text are not playing. So what we're going to do for questions is Cameron's going to moderate, and we can take them from the ASM queue room as we collect some things over Twitter. And while everyone's adjusting their screens and mics, what we can do is ask the first question, which was just to confirm, are these all complete genomes that you're dealing with, or any of them partial, and would that affect any of these analyses?
They're as complete as we get with a lot of these. We're not closing every gap, but when we use tools like Segma, which is a tool for assessing the genome completeness by about 250 or so conserved proteins, we get numbers in the 90s or 95% completeness for most of those. So I think we're okay. That HP genome is not complete. I mean, it was sort of it's probably more in the upper 80s, but we recently did another sequencing on that, and we're able to improve it. I think the numbers still, the patterns still hold up in terms of the recent changes.
Cool. Are there any questions from ASMQ? If not, I have a question about just dealing with some of these animal pathogens. You got into the frog pathogen. I was wondering if you could comment, just for general purpose, on the bat pathogen. Isn't that also a fungus, and how does that fit into some of these things that you've been discussing?
Yeah, so I mean, I give this lecture in my class about sort of emerging infectious disease. There's a lot of new fungal pathogens, or pathogens that are causing new problems. So yeah, Pseudogymnoascus destructans is the white-nosed bat pathogen, and it's an ascomycete fungus, so it's different from these. It's unfortunately, it's kind of slow growing, so it's kind of a pain in the lab, but it's definitely, you can culture both of these without the host. It seems like there are protease changes as well in that organism, from what I've heard. It's got expanded proteases, and so that potentially could be another route. I think we're seeing it over and over again, that protease, when things become animal associated, they seem to have different proteases, or at least more proteases that are needed. And when we see things that were plant saprotrophs, they typically have a lot of other plant matter degrading genes, and we see them lost. When we compare things like coccidioides, it lost a bunch of genes that its relatives have that are plant saprotrophs. So yeah, I don't know if that answers your question about pseudogenesis.
Yeah, just sort of a follow-up. We talk a lot about when we're dealing with, and we've seen some examples of this on microseminar of like obligate intracellular symbionts that are bacteria. Are some of these lifestyles associated with genome reductions, like massive shrinkages of genomes, or maybe examples of even something like streamlining or anything like that?
Definitely the microsporidia have become extremely streamlined. So that's, and they both have smaller genomes that are streamlined, and they seem to have faster rates of evolution. So their branches are super long, those guys. For Rosella, it doesn't seem like it has reduced its genome in the same way. It seems to still be a nicely behaving genome relative to the other ones. So we don't see that. And does that mean that maybe there's more of a cryptic non-host associated lifestyle? Is that what it is, or has it just not specialized to that point? I guess that's the question. For BD, like the frog pathogen, there's evidence that it might have other reservoirs in the environment, so it might be that, again, that it's really good at killing the host, but it has some other place that it also has to live, and so therefore it can't afford to streamline because it still needs those other free-living factors.
Yeah.
I know that, like, John McCutcheon's looked at this, and obviously in the bacteria, but he's also looking at it in some of the insect-associated fungi, and they're not, without giving away too much of their stuff, I don't think they're not seeing the same extreme reduction in streamlining when things become host-associated for their examples. So
yeah, I wonder if that's an evolutionary time thing, right? Like, I mean, fungi and insects are nowhere near as distantly related, I guess. Although I wonder how long it takes, or maybe it's just a matter of how big you're starting with and how much you can drop off.
Right. So I guess it's like, how long have you been associated with the host, and what percentage of your life has to be, or how obligate are you?
Yeah.
Because that, maybe those would be factors that would better predict the degree to which you see streamlining. I'm guessing that would be the case. I mean, there are things that are commensal, like Candida, or things that are commensal with animals that are really mostly associated, and they have smaller genomes, but I think it's more because they're part of the yeast group that have smaller genomes, not because they've reduced to the host association. But that's a really important question. I think we're going to have other examples on the tree where people are going to tackle that. So I hope we'll get answers.
Okay, well, we've got 5 minutes left, so if we can ask anyone at ASMQ to come forward, we'll turn on your mic. You can ask a question directly, or feel free to ask us on Twitter. I haven't seen any other questions come in through Twitter, but just wanted to actually interrupt you guys to make sure other people got a word in if they needed to.
Of course.
Yeah. So we will hang out for 1 more minute, asking ASMQ to jump in whenever you guys need to, or if the bond group is still there and has any questions. Well, in my dead air time, I'll ask my question actually, which is, we see, I don't know if you've ever read our papers, but we see fungi in these deep sediments, right, dealing with all sorts of extreme pressures. Our colleagues have been able to recover them and grow them. They seem like they're legitimate, and they're not just in sporulated forms. We've recovered mRNA from them. And my throwaway, right, is like, you're kind of ruining me with these 1,000 genomes. Because my throwaway is always like, oh, there's more diversity than we know. But if you're really able to define sort of the increase and decrease in terms of these cell structures that are needed, or different ways that you can see different life forms, right? I'm thinking about the dictyostelium example in small eukaryotes, where you have a bigger genome because you have different multicellular forms. But how much space do you think is there outside your Noah's Ark in that genome space?
Yeah, I mean, so are you saying, are there things that we put some boundaries on what we call fungi, and do we think that everything we're going to find now is sort of within those boundaries, right? That's partly the question. Yeah, I mean, I guess if we talked about this 5 years ago, we wouldn't have said anything about Cryptomycota, so we wouldn't have thought that was there, and that seems to be as diverse as some of the fungi. So I mean, is it this, sort of, is it a shadow clade that's totally diverse, as diverse? But then it's weird, if lots of them are parasites of other fungi, then how did they become so diverse? When did their host come about? So it might be that they have had alternate lifestyles, and we're just sort of seeing the echoes of that. I think there's more to be discovered as we go deeper in the sampling of places. So it might be that there's another, branch that we haven't really been able to place things on. But it might fall within what we've already called the boundaries. But it might still be, in terms of it evolved from an ancestor that we've already kind of know about, but there may still be other branches that are quite different. And so, I'll stop if anybody has a question.
I was going to say, actually, we are out of time.
Okay, out of time. All right.
I am going to stop the live broadcast, but we can still chat inside the Hangout if needed.
Okay.
I'm going to hit stop to YouTube. Thanks, everyone.